Hey, and so we're back. Now, again, the basic idea of cellular respiration is you're taking food, stripping away the electrons and their associated protons from that food, and then using that to power the production of ATP. So, this process of taking the electrons from food and then using it to make ATP, it's broken down into three sets of chemical reactions. And these reactions occur in different parts of the cell. Now, if we look at this basic diagram here, the first of those three processes, glycolysis, that happens in the cytosol of the cell, so in the sort of fluid part of the cell. And then the last two sets of reactions, the Krebs cycle and the electron transport chain used for oxidative phosphorylation, those happen within the mitochondrion. Okay. So ATP production happens in three sets of reactions: glycolysis in the cytosol, and then the citric acid or Krebs cycle, and then oxidative phosphorylation, which uses the electron transport chain within the mitochondrion. So sort of keep that in mind as we go through this. So as we talk about glycolysis, keep in mind glycolysis is happening in the cytosol or the fluid part of the cell, not in any particular organelle. All right, so with glycolysis, what the heck is going on here? Well, let's use the name to help us sort of introduce the idea. Gly means sugar, lyse means to split. So in glycolysis, we start breaking down sugar. Now, something to keep in mind, the College Board loves to ask glycolysis questions. Here's the deal. Glycolysis, it happens in the cytosol, right? Not in any particular organelle. You have everybody doing it. So we do it, plants do it, fungi do it, prokaryotes do it. Anything that's alive is performing glycolysis. So if you have this process that everybody's doing and it doesn't require any organelles, it doesn't even require oxygen, right? Or like molecular oxygen from the atmosphere. So if you got this really old process, or if everybody's doing it and you don't need any special organelles, you don't need atmospheric oxygen, that all suggests that this is a really old process. A lot of times in the AP exam, they ask questions about, you know, organisms that perform glycolysis or the age of glycolysis. It's all linked to the idea that it's shared and it doesn't require any special organelles, so it's something that's been around. So this is one of the first metabolic pathways. Okay. Now, what happens during glycolysis? In the College Board curriculum, there's a lot of emphasis on inputs and outputs. So what goes in, what comes out. So if we follow the arrows here, you start with glucose, this 6-carbon molecule, 1, 2, 3, 4, 5, and 6. And when it's done, what do we end up with? A couple of pyruvate molecules. And if we look, each pyruvate is 1, 2, 3 carbons long. So in essence, you take a 6-carbon glucose and you break it into 2 molecules of 3-carbon pyruvate or pyruvic acid. Okay. Now in this process, you do strip away some protons and electrons. So that's how you form this NADH. So with glycolysis, we strip away some protons and electrons from glucose. In the process, you net a couple of ATP. So what do we get out of glycolysis? You get a couple of ATP, not enough to live on. You get a couple of NADH, which are electron carriers. And then you get a couple molecules of pyruvate. Okay. So at this point, most of the energy from glucose is still trapped in pyruvate. So you can break this down further and extract more energy out of it. And that's exactly what happens when you move inside the mitochondrion. So in the cytosol, again, you take glucose, you run it through glycolysis, you strip away some protons and electrons to form NADH, you make a couple of ATP, but most of the energy is still locked up in these molecules of pyruvate or pyruvic acid. Now, what happens next is a reaction called the link reaction. So here in the cytosol, we have the pyruvate molecules, the 3-carbon pyruvates. When that is transported into the mitochondria, the matrix of the mitochondrion, look at what happens. One of the carbons is stripped off and forms CO2. When that happens, you're left with this 2-carbon molecule, acetyl coenzyme A. Now, you don't necessarily need to recognize the name of acetyl-CoA, but just recognize that it's a 2-carbon molecule, okay? So again, what's happened is you take your 3-carbon pyruvate, send it inside the mitochondrion, it goes through this link reaction, and you end up making a 2-carbon acetyl-CoA. Now, one of those carbons that got stripped away, again, it forms CO2, so you exhale it. And in this process, you strip away a proton, electron, and you form an NADH. Okay? So now, after the link reaction, we have this 2-carbon acetyl-CoA. Well, this is where acetyl-CoA enters the Krebs cycle, or the citric acid cycle. Now, this is something where, again, focus on inputs and outputs. There are a lot of steps here. If you want to be a doctor and you're going to take the MCAT, you can memorize all these steps. But for AP Bio, hey, just focus on the big picture, right? What's happening overall? So there is an existing molecule, this oxaloacetate. Again, don't worry about remembering it. But what happens is acetyl-CoA joins to it, and it forms this citrate molecule. Now, citrate, it'll undergo a number of different reactions, and the gist of the idea is when you go through all these reactions, what ends up happening is you strip away protons and electrons, so you form NADH and FADH2. Those are the electron carriers. So again, you're stripping away protons and electrons. You'll net a couple of ATP out of this. So again, not enough to live on. And what happens is you strip away the carbons and it forms carbon dioxide. So you have the 2-carbon acetyl-CoA. The carbons will actually get stripped away and converted to CO2. So, the pretzel that I ate, some of the carbon can be used to make tissues of my body, but the carbon that enters the mitochondrion, you're going to end up exhaling it. Okay, so it's sort of crazy to think about, but what we eat, some of it will become part of our bodies, but what doesn't become part of our bodies, we got to get it out, right? And the way we get rid of it is by exhaling it as CO2. All right. So again, you've taken the glucose from your food, you've broken it down during glycolysis into pyruvate. Inside the mitochondrion, the link reaction takes that 3-carbon pyruvate and turns it into 2-carbon acetyl-CoA. And then acetyl-CoA enters the citric acid or Krebs cycle. In that process, you strip away some protons and electrons and form NADH and FADH2. You take the carbons, split those off, and release those as CO2. So exhaling breakfast, right? And you get a couple of ATP out of it. All right. So at this point, we've taken all the carbon from glucose and got rid of it. We've exhaled it as CO2. We've only netted like 4 ATP out of this process. That's not enough to live off of. So what the heck have we done? We've stripped away a lot of protons and electrons, and we've made a fair amount of NADH and FADH2. So what are we going to do with that? Well, that is what we do in the third set of reactions here. That's oxidative phosphorylation. So what do you do with all that NADH and FADH2? Well, you just send it to the Christie. If you'll recall, mitochondria have 2 membranes, that outer membrane and then the highly folded inner membrane. That highly folded inner membrane is called the Christie. And here it's zooming in on the Christie. Okay, so this is really zoomed in. Let's jump back just a little bit. Here is the outer membrane. Here is the inner membrane. Here's the fluid inside the mitochondrion. It's called the matrix. And the space between the outer membrane and the inner membrane is called the intermembrane space. So jumping ahead, this might be nice. Here you go. Outside the mitochondrion, here is the matrix. This is the Christie. And here's the intermembrane space. So now we're going to zoom in on this quite a bit. Here's the Christie, that inner membrane. This is the intermembrane space, and then that's the matrix that's in the middle. So what happens? If we look, there's the NADH and FADH2 that have those electrons and protons from our food. So again, through glycolysis and through Krebs, we strip away protons and electrons and put them in these carriers, NADH and FADH2. Now, in the third set of reactions, oxidative phosphorylation, we use proteins in the electron transport chain, ultimately to make ATP, lots of ATP. Okay. Now, just to sort of get the lay of the land, embedded in the Christie, embedded in that membrane, are these proteins of the electron transport chain. Those are the purple proteins here. And then there are little shuttles in between them. Okay, these are other proteins. A lot of times they're called cytochromes. okay, or they're cytochrome proteins. So there are these proteins in the cristae that form the electron transport chain. And then there are these little shuttle proteins that carry electrons to and fro, these different proteins. Now, if electrons are moving from here, protein complex 1 to 3 to 4, you can see that, or you can guess that these proteins are increasingly electronegative, right? If the electronegativity is greater and greater, then the electrons are going to get passed along. Now, the electrons get shuttled down the electron transport chain here. But what happens with the protons in NADH and FADH2? Well, those protons get pumped into the intramembrane space. This is a process called chemiosmosis. Chemiosmosis is not about water, it's about the movement of ions. Okay? So what happens is you create this gradient, and through the process of chemiosmosis, these protons are going to flow down their electrochemical gradient. Rather than water flowing down its concentration gradient, protons flow down their electrochemical gradient. And then that powers the enzyme ATP synthase to make ATP. Whew. Okay. So let's look at this a little more closely. So here, embedded in the cristae, are the proteins of the electron transport chain. NADH and FADH2 bring the protons and electrons from our food, and they pass off the electrons to the electron transport chain. Now, again, here's like cytochrome C, they're the little shuttles. The proteins complexes in the electron transport chain become increasingly electronegative, and eventually those electrons have to go somewhere, right? So something has to accept those electrons at the end of the electron transport chain, and that something ends up being oxygen. Why do we need oxygen? We need oxygen for this process, okay? So what happens is we breathe in oxygen, send it to our cells, have it go inside the matrix of the mitochondria. And then when electrons get passed down the electron transport chain, oxygen accepts those electrons. Now, it also needs to combine those electrons with something, right? So what happens is the protons that got pumped into this space flow through ATP synthase, and then the protons combine with the oxygen to form H2O. So when we breathe in, that oxygen we breathe in is going to combine with the protons, and it's going to form water. Okay, so it's sort of crazy to think about, but again, you got to do things with the molecules that are in your body. So we breathe in oxygen, have it combine with protons that got passed through ATP synthase, and then it forms water. So the air we breathe in eventually gets made into things like the water we exhale or the urine we produce. Okay, that's pretty cool. So that's the basic idea. The protons and electrons from our food get passed along the electron transport chain and get used to make this gradient. When the protons flow down their electrochemical gradient, it makes this enzyme ATP synthase spin. When ATP synthase spins, it brings together ADP and phosphate, and then it forms ATP. Okay, so we can get something like 32 ATP per glucose. So when you break down a glucose molecule, numbers vary based on sort of the efficiency of chemiosmosis, but you can get about 2 ATP from glycolysis. So again, with the breakdown of one molecule of glucose or 1 mole of glucose, you get 32. You get 2 ATP from glycolysis, 2 from Krebs, and about 32 from electron transport. So this is the big payoff here. All right. I think that hits a lot of the highlights. This again just sort of runs through the idea of chemiosmosis. You strip away the protons and electrons from NADH and FADH2. The electrons get passed down the electron transport chain. The protons form this gradient. When they flow down the gradient, that's the idea of chemiosmosis, an ion flowing down its electrochemical gradient. It spins ATP synthase that brings together ADP and phosphate, and boom, you got ATP. And once you have ATP, then you can power all sorts of chemical reactions, and you got life. All right, so let's sort of tie it into the broader picture of what the heck is going on. So here's the idea of cellular respiration. This is what we just talked about. Look at the reactants in this equation. You need glucose, you need oxygen. What do we end up making? We make CO2 that we exhale. We make water that we exhale. And then we get some ATP out of this. All right, now let's jump into the next chapter or look ahead to the next chapter. Photosynthesis. What does photosynthesis do? It takes carbon dioxide and water. Using the power of light, it makes glucose and oxygen. So these are not reverse reactions. Definitely don't get that idea. Sometimes they ask something like that on the AP exam where they get people to think that photosynthesis and respiration are the reverse of one another. They're not, but they are tied to one another, right? Um, for cellular respiration, we need glucose and oxygen, and we produce carbon dioxide and water. Photosynthesis, it requires carbon dioxide and water, and it makes glucose and oxygen. So when I think about who needs whom, what of these processes can we do? We can perform cellular respiration, right? We can take our food, break it down, take the oxygen we breathe in, and make ATP, but we're going to make these waste products. Now, what can plants do with those waste products? They can use it to actually make more food and oxygen for us. Now, the thing is, plants and other photosynthetic organisms can do both these reactions. They can perform photosynthesis and cellular respiration. All sorts of organisms have mitochondria and chloroplasts. We are completely reliant upon photosynthetic organisms for the continuation of life. If we didn't have photosynthetic organisms, we wouldn't have the food we need, and we wouldn't have the oxygen we need. So we are wholly dependent upon the photosynthetic organisms of Earth, because without them, we wouldn't be able to make the food we need or breathe the oxygen we need. So really pretty spectacular. So it's cool. You can take the foods we eat, turn them into gases, and then photosynthetic organisms can take those gases and turn them back into food for us. So it's really a remarkable cycle that we're involved in, and it's just really cool that we get to understand components of that. All right, so that gets us through the big part of how we turn food into ATP, or use food to make ATP, I should say it that way. And then we have just one more segment left, looking at other ways to make things work. Thanks a lot.